Sound. You are getting a broken sound.
Yeah.
Yeah, please adjust it. Yeah.
Broken voice.
We welcome all distinguished guests for this program. I must acknowledge all participants for this overwhelming response. I welcome all academicians who have registered for this expert talk on vaccine development, the current pandemic scenario. I heartily welcome all respected faculty members and all participants from various colleges. So now move ahead. I request Dr. Pradnya Bhalirao, convener of Popular Lecture Series, to put her welcome speech. Over to you, madam.
Thank you. Thank you very much. Thank you, madam. Good morning, everybody. And first of all, I welcome you all for this science event organized by IVSA. I welcome all the management members of Banzari College, Banzari sir, Banzari madam, and all the president, secretary, principal of Kamala Nehru College, the staff and students, and Dr. Anuradha Gadkari, our torchbearer of IVSA, and all my executive members, and all the IVSA members and other members who are present here. And I am really, I'm really delighted to see the students here. Most of the students who, huge number of them have joined this activity in science, which is organized by a scientific body of Nagpur. Friends, vaccination is a deliberate attempt to protect humans against the disease. Vaccination actually dates back to 18th century, and in 19th century, vaccinations were prepared in the laboratories. It were developed in the laboratories. In 20th century, vaccinations based on immunological markers were developed in the labs, of course. And now, in 21st century, we have the vaccination involving molecular structure of the cell. You must be reading about the vaccines today, which vaccines are developed, and RNA vaccine. So what is very special about this, that when RNA, once it is fired from the nucleus, it never goes back to the nucleus. So there are hardly any chances of interfering this vaccine with the DNA of the receiver. So therefore, this vaccination is important. And actually, vaccination is the most effective medical intervention to control the disease. As I understand, epidemiology and social science go hand in hand, because they say that there are going to be more and more epidemics in future. We might get, because of this climate change, environmental changes, changing ecology, urbanization, and more people are traveling now. So it will become more complex to control a disease or the spread of the disease. So today I'm not going to be in more details, because we have invited Dr. Shalini Chahande to elaborate on this topic. And let me introduce you, friends, to IVSA, Indian Women Scientists Association. It was the body formed by some scientist women way back in 1973, and soon this association came to Nagpur by the efforts of Dr. Anuradha Gadkari and her friends, who brought IVSA to Nagpur. Now we have 38 years, we are 38 years here, and for all these 38 years, we have been trying to popularize science and to reach the science to the society, to masses, to classes, and to each and every person of the society. And we try that how it can be done. So we organize programs like this lecture also, like quiz, like other competitions, like poster competitions, seminar competitions, and now these webinars. And through these, our more goal is to popularize science. I don't want to go in details because I have a huge list of my programs. So this is not the platform to narrate those. Welcome you all again. And I hand over now the proceedings to the organizer. Thank you very much.
Thank you, ma'am.
May I add something to this? May
Yes,
I
sure.
add something?
Yeah, sure.
Yeah. We have an intercollegiate event for the college students every year.
Yes.
And this event today, we are celebrating at USA as intercollegiate event, wherein 4-5 colleges have joined. It's usually college students joining from different colleges, registering with us, and we do some activity for the college students.
Yeah.
The aim of IFSA target group is general people, and in that we very happily and very proudly include college students. So please clap. This is an intercollegiate college event.
Thank you, madam.
Thank you, madam.
Over to Shalini. Over to Shalini.
Thank you.
Hello, ma'am. Can I start? Yes, ma'am. Sure.
Just a minute, madam. Just a minute.
Yeah.
Thank you, Dr. Palero, madam, for giving us the insight on the objectives of this expert talk. Now, I request respected principal sir, Dr. Diabetic, to deliver an introductory remarks. Over to you.
Thank you, ma'am. Good morning, all of you. First, I pay tribute to visionary leader Let's Govindaraoji Vanjari, founder president Amar Samandal. On the behalf of Kamla Nehru Mahavidyalaya, I, Dr. Dilip Badwai, principal of the college, extend a warm and heartiest welcome to all respected guests, faculty members, and students of different colleges on the occasion of inaugural function of webinar on vaccine development, the current pandemic scenario, organized by Kamla Nehru Mahavidyalaya in association with Indian Women Scientists Association, Nagpur branch. Speaker for this webinar, Honorable Dr. Shalini Chahande ma'am, Assistant Professor, Department of Biochemistry, Porwal College Committee. Convener of this webinar, Dr. Sandhya Bhalerao ma'am. Webinar will be summarized by Dr. Anuradha Gadkari, Somalwar ma'am, other respected members of IUSA, our faculty members, Moge madam, Shardul Wag sir, Neha Thakur, other faculty members, and my dear students. Once again, I welcome you all with my words. Regarding the IUSA, ma'am have already explained. Few women scientists came together, form a body named as Indian Women's Scientists Association. There are in all 11 chapters, including Nagpur also. And main aim of the IUSA is to popularize the science by conducting different events for the students. Hence, we are facing the challenges due to COVID-19 pandemic since month of March 2020. Since last few months, government started Mission Unlock, phase 1, 2, 3, with certain guidelines to be followed. Government granted permission to open schools in the month of December 2020 and colleges from 15th February 2021. Immediately, corona cases increased exponentially, and again government ordered closing of school and colleges. We the people are responsible for increasing corona cases. We are not wearing masks, not keeping social distancing, wandering here and there without any work. Dear friends, being responsible citizen, we have to follow the norms issued by government and ICMR to keep corona away. During this pandemic situation, we came to know the importance of vaccination. Vaccines can prevent infections, infectious diseases. Example of vaccine preventable diseases are measles, polio, hepatitis B, influenza, Corona, we are now know many others. When most people in the community are vaccinated against the disease, the ability of such viruses and bacteria to spread is limited. This is called herd immunity. When many people have immunity, this will indirectly protect the people who cannot be vaccinated. During November and December 2020, corona cases showed decreasing trend. Then we feel that corona is under control and herd immunity developed in the society. Government also carried out zero survey all over the India in Nagpur district too. Its result shows that around 50% of the peoples developed herd immunity in Nagpur district, and we became careless, started attending marriages, social function, marketing and whatnot. Results we are experiencing now. There is increase in the corona cases. Vaccines greatly reduce the risk of infection by training the immune system to recognize and fight against the virus and bacteria. Vaccines safely deliver an immunogene, which is a specific type of antigen that elicits an immune response to train the immune system to recognize the virus when it is encountered naturally. Vaccination against coronavirus started in India in the month of January 2021. Initially, corona warriors like health workers, frontline workers, then for the remaining peoples having the age of 50 years with severe diseases. Now, corona vaccine is available for all common peoples also. Our college is importing not only quality education, but also arranging various activities for overall development of students. We are updating our students with current development in sciences. This results into today's webinar on vaccine development, the current pandemic situation. Under the dynamic leadership of Honorable Dr. Vanjari Madam, President Amar Seva Mandal, Honorable Advocate Abhijit Vanjari Sir, MLC, Maharashtra State, and Secretary Amar Seva Mandal, Honorable Dr. Smita Vanjari Madam, Treasurer Amar Seva Mandal. Our college each continue to attain new height. As a result, Kamla Nehru Mahavidyalaya is re-accredited A+ grade in its third cycle of re-accreditation with CGPA 3.53. We offers 9 graduation courses, 22 PG courses, 2 PG diploma, 6 higher learning and research centers that includes physics, chemistry, electronics, computer science, commerce, English. 30 students are already registered for PhD from our center. We are imparting education to around 8,000 students. I hope students and faculty members will definitely be benefited by this webinar. I wish great success for this webinar. Thank you very much. Over to you, ma'am.
Thank you. Yes.
Thank you,
very
sir.
much, sir, for your remarks. Now, we have an eminent speaker with us, Dr. Shalini Chahande, for today's expert talk on vaccine development, the current pandemic scenario. Now, I request Dr. Sandhya Moghe Madam, Head, Department of Biotechnology, Kamla Nehru Mahavidyalaya, to introduce our today's guest speaker. Over to you, Moghe Madam.
Good morning. I feel pleasure to introduce our today's chief guest, Dr. Shalini Chahande Madam. Madam is doctorate in biochemistry from RTM, Nagpur University, Nagpur. Presently, she is working as assistant professor, senior scale in the Department of Biochemistry, SK Porwal College, Kamptee. Recently, Madam is awarded with a Bharat Ratna Dr. Radhakrishnan Gold Medal Award for excellence in her field on the occasion of 73rd National Unity Conference on Individual Achievement and Nation Development on 22nd Feb 2020 in Chennai. Madam was awarded Junior Fellowship by ICR in the subject of biochemistry from 1993 to 2004 program. Madam also awarded GRF Fellowship by UGC in Science and Humanities. Madam has presented her research paper in the International Conference on Current Trends in Biochemical Research and Education. Madam has presented research project at ACBI Millicon Annual Conference. Madam serves as contributory, ad hoc, and regular teacher in the various colleges like Department of Biochemistry, PGTD Campus, RTM Nagpur University Nagpur, Kamla Nehru Mahavidyalaya, Department of Biochemistry, Nagpur, GH Rai Soni College, and College of Agriculture and Engineering and Technology, PDKV Akola. So we all are waiting for such a dynamic lady to over to you, Chahande Madam.
Thank you very much, ma'am. A very good morning to all of you. I extend my gratitude to the Management Committee of Kamla Nehru Mahavidyalaya, Nagpur, Principal Dr. Badwait sir, distinguished personalities of ILSA, Convener Dr. Pradnya Bhalerao ma'am, Dr. Seema Somalwar ma'am, Principal Somalwar Nicholas Mahila Mahavidyalaya, Dr. Anuradha Gadkari ma'am, founder member of IFSA, for giving me this opportunity to deliver this lecture. So friends, let us see or go into detail about what the vaccine development is. Let me share the screen. Myself, Dr. Shalini Chande, and I'm going to speak about vaccine development, current pandemic scenario. Now, today I'm going to cover these topics: how the immune system works in brief, what are the vaccines, and why do we need the vaccines? Brief history of vaccines, type of vaccines against coronavirus, vaccine development which has taken place till now, vaccine storage, India and COVID vaccine, and what is COVAX. So we should dive into this particular matter and see what are the intricacies about this particular coronavirus vaccines. Now, the world can't return to normal without safe and effective vaccine against the SARS-CoV coronavirus. Along with the coordinated global vaccination program, we can come up out of it. Now, I'm talking about the vaccine. So what the vaccine does? Now, the vaccine prepares a person to fight off an infection by training the immune system. Now, we must be wondering, what is this immune system? So for the layman, it is very difficult to understand the immune system in detail, but I'll make an attempt to explain few aspects about the immune system. So it's very useful to know how the immune system works in order to understand why vaccines are important and why we are all waiting for one against the novel coronavirus. So we are waiting, and some of them have got their jabs till now. Now, how the immune system works? Now, defenses against the causing germs or the pathogen, whatever the pathogen enters in our body, what happens? Our body prepares us in two ways. The first thing is we get the innate immune system, and in another way we get the adaptive immune system. Now, what is this innate immune system and what is the adaptive immune system? Innate immune system is the first line of defense which we have in our body, and in this innate immune system, it responds by detecting the molecular pattern characteristics of a microbe. What is the molecular pattern of that particular microbe? Like the sugars on the bacteria, or whatever the structure of that particular virus is. So whatever the infection we have got, so our body recognizes it first in the arm of innate immune system. While in the adaptive immune system, we learn to recognize, our body learns to recognize and remember the specific pathogen. So together they create the immunity and we are protected from the infection. Now, we'll go in detail with the innate immune system, what it does in case of corona also, what it does to the body. So this is the first line of defense, as I already told you. Now, whenever we encounter the pathogen, now in this case, a virus. So in this innate immune system, we have various barriers like the skin and other molecules like the antiviral interferons, which are secreted by the cells, and the white blood cells, which we call as the leukocytes. Now, the innate immune system, it responds immediately as soon as the virus enters in the body, deploying its weapon to the site of infection. Now, what it does, whenever the infection comes inside your body, the inflammation occurs. Now, when the inflammation is there, some white blood cells act on as a guard. Now, they guard us to watch out for the invaders and certain types such as the macrophages and the dendritic cell. So, along with the leukocytes, we have the macrophages as well as the dendritic cells. They will eat and digest the pathogen through the phagocytic process. So, phagocytosis is very important in our body, and it is always there. It's a natural process which takes place in our body. So, now the research in this COVID, what it suggests is innate immune response to SARS-CoV is similar to, the coronavirus is not yet been studied in detail till now. Researchers are still searching for the ways how it infects and what are the strategies to overcome this particular. But what we have till now studied is the close relatives of this virus. And the close relatives of this virus are what? SARS-CoV-1 and MERS. So these two viruses we know, which causes the severe acute respiratory syndrome and the Middle East respiratory syndrome. We already know. And this outbreak, it took place in the year 2002 to 2003. During that time, we came across these viruses. So,
Yes,
like its relatives, SARS-CoV-2 suppresses the immune system. Now, if the immune system is suppressed, what will happen? So, when the immune system is suppressed, it disrupts the immune cells and the interferon molecules, because we are protected by the immune cells as well as the interferon molecules. And what the virus does? The virus suppresses these. So that immune suppression ability might be the reason why COVID-19 has such a long incubation time. See, whenever someone is being infected with the coronavirus, we don't really get the symptoms very easily. It takes days to have the symptoms. And sometimes the subject goes asymptomatic. But when it goes asymptomatic, it doesn't mean that the person is not having the coronavirus infection. So it usually takes 1 to 14 days for the infection, or 1 to 4 days for the infection to go. So immune suppression probably explains why coronavirus is able to wreak havoc on the innate system, recruiting too many white blood cells. Now what happens whenever the innate immune system is active? What will it do? It will activate all its guards. And the very potent guard is what? The white blood cell. What will this white blood cell do? Because this is the respiratory disease, this white blood cell, all the cell, they will go to the site of infection. So they will go towards the lung, and there they will cause excessive inflammation. And the inflammatory response might in turn lead to acute respiratory distress syndrome and causes shortness of breath, lung injury, and severe cases of coronavirus disease. So in this way, we are affected because of the innate immune system, which is protecting us, and what the coronavirus is doing to the innate immune system. So how the coronavirus is disrupting our innate immune system. Now we come to the next system, and that is the adaptive immune system. Now in the adaptive immune system, we have two players, the B cells and the T cells. Now in this particular B and T cell working for the adaptive immune response, our body is adapted. Now what will it do? Now antibodies will be, it will take some time. Whenever the infection, the person is infected, the virus goes inside the body takes its time, the B cell, they become activated, the T cell, they also become activated, the antibodies are generated. Now the antibodies which are generated, that match and bind to the antigen molecule, and that are produced by the B cells, and anything covered with a specific antigen, so such as a virus particle, and once attached to the matching antigen, antibody might physically block the microbe from invading your body. So it will do that and allow the invader to be more easily ingested by the phagocytes. So antigen antibody complex will take place and it will be ingested by the phagocytes or mark it for destruction by the enzymes. Another system which comes into play in the body, and that is the complement system. So in the complement system, whenever there is the formation of antigen antibody complex, this complex is now destructed or it is removed from the body by a system of complement proteins. So these are the executioners. Other executioners like the natural killer cells are also there, which are protecting us. So COVID-19 test can detect antibodies within 1 to 2 weeks after the infection. So the person develops the symptoms and we get the certain type of what we can say illnesses. Now, what the T cell do? B cell, we know that the work of B cell is to produce the antibodies. And another thing is there that B cell also has the memory, very important thing, which the B cells has. Now, what the T cell do? For following invasion by the pathogen, the dendritic cell ingests the germ's antigen and travel to the lymph node. So in our body, the lymph nodes are located all over the body. So there, what they do, they do their job as professional antigen presenting cells. Dendritic cell will be acting as a antigen presenting cells. Now, and show the foreign antigen to the T cells. So they will show to the T cell. Now, once activated, the T cell will divide and circulate in the bloodstream. So they will divide and they will be circulated to the bloodstream and ready to recognize and fight any invaders that is encountered in the process. So in this way, the T cell recognizes the foreign antigen. So, but when it's presented on the surface of an infected cell, they come in two types. The two types of T cells are the helper T cells and the cytotoxic T cell. So what are the functions of the cytotoxic toxic T cell and the helper T cell? The cytotoxic T cell that kill the microbe or the virus infected cells, infected body using the destructive molecule, and the helper cell aid the other immune cell and the B cells to generate the antibodies. So in this way, the T cells also work. One more thing is very good in this case, and that is the T cell, they have the memory. So they remember if the infection is, if the person is reinfected again. So in this way, the immune system works. Now, what are the vaccines and why do we need the vaccines? This is a very important thing to know. Now, everybody knows, but still I would like to elaborate what the vaccine is. The vaccine is the product that stimulates a person's immune system to produce immunity to a specific disease, protecting the person from that particular disease. So we want to get rid of corona. So vaccines, we need the vaccines against it. But the mode of delivery of these vaccines are like, it can be administered through injection, or it can be administered by mouth or sprayed in the form of nasal sprays. Now, vaccination and immunization, both the things are interchangeably used. What is vaccination? Vaccination is nothing but the act of introducing a vaccine into the body in order to produce immunity to a particular disease, a specific disease. While vaccination is a process by which a person becomes protected against a disease, and that is through vaccination. So vaccination is important for immunization. So therefore, we interchangeably use these two words. Now, a brief history of vaccines. Now, we should know from where the vaccines have come. Now, the first vaccine developed was against the smallpox by Edward Jenner, a physician, English physician in Berkeley. He found that dairy maids with cowpox were relatively immune to the cowpox. So what he did, he took the exudate and secretion from a cowpox pustule on the hand of a dairy maid, Sarah Nilbs, and inserted it into the arm of an 8-year-old boy, James Phipps, on March 14, 1796. So in the year 1796, he did this. He did this experiment with the 8-year-old boy. The vaccination was effective since the boy did not catch smallpox even when he was infected with smallpox virus. So 6 weeks after the vaccination. So the boy, he was infected, but he was not ill because of the vaccination. At that time, it was not known that Edward Jenner has done the vaccination. He was just doing an experiment, but hence it was proved that it is working as a vaccine. So Jenner published his findings in 1798, and controversies rose during that time, but slowly and progressively it was accepted by all that vaccination is there. Now, why is vaccination so important? Now, this I have already explained, so I'll not go in detail. Now, herd immunity. Now, everyone is well versed with what is herd immunity. Everyone knows what is herd immunity and all. So this herd immunity, when the herd immunity is very important, especially who scientists, the chief scientist Somaiya, she says that when the majority of the population has been vaccinated against a disease, she has given the example of the measles, where 90% of the population, if it is vaccinated, then that particular disease will eradicate. Now, in case of coronavirus, what will happen? There will be a few people left whom the infection can spread. If we vaccinate 60 to 70% of the people, still 20 to 25% people will be there who are not vaccinated. Why those people are not vaccinated? Different reasons are there. Like some people, they don't believe in vaccines. This is one reason, but this reason is not acceptable. Another reason is people who are having weak immune system. If the people are having the weak immune system, then what happens? The immune system reacts in an erratic way in those people. So it is very difficult to administer the vaccine and see the consequences. So we are not going to see the consequences, what is going to happen. And in some elderly person, those who are having some comorbidities, and if the vaccine is administered, what is going to happen? So in these many people will be left over from vaccination, and for them, what we need is we need a herd immunity. So another, see, the herd immunity is one approach. 60 to 75% people are vaccinated, then the other 25% of people, they will be protected from this particular infection. This is one thing. Another treatment approach for the non-vaccinated individuals is, now what happens? The body makes the neutralizing antibodies. Now what happens? Whenever any antigen goes into the body synthesizes antibodies against it. Now, if the immune system is strong, then during that time, what happens? A strong immune system develops in your body. And because of the development of a strong immune system, the neutralizing antibodies are developed. Neutralizing antibody mean the antigen and the antibody will react and it will be neutralized and it will be taken out from the body. So neutralizing antibodies that specifically target the receptor binding domain of the SARS-CoV-2 spike protein. We know how the coronavirus infects us. The coronavirus infects us through the spike protein. The spike protein goes and sits on the ACE2 receptors. Where are these ACE2 receptors located? They are receptors. These receptors are abundantly present in the lung alveoli, as well as there are many cells in our body where these ACE2 receptors are present. So this spike protein of the coronavirus goes and sits there, and then the cascade of reaction starts. So the spike protein are thought to be essential for controlling it. So the important target in the treatment will be the spike protein. Second approach. First approach is the herd immunity. Second approach is the spike protein. So the majority of the people can produce neutralizing antibodies against the novel coronavirus in severe case of COVID-19. So according to a study that supports the use of combination antibody theory to prevent and treat the disease. Now, this we have already seen that in many patients, passive immunization is done. What is passive immunization? If a person who is contracted with the coronavirus disease and that person has developed the antibodies, then the plasma is taken from that particular person and the plasma is administered to a person who is already infected with the coronavirus and who is having the severe effects of the coronavirus. So this is the work of the antibodies, neutralizing antibodies. Now, researchers noted that the neutralizing antibodies that specifically target the receptor binding protein, the receptor binding protein domain of the SARS-CoV spike protein, which the virus uses enter the human cell. Now it enters the human cell as thought to be essential for controlling it. Now what this receptor binding protein do? Neutralizing antibodies have been detected in convalescent patients, those who have recovered from COVID-19. So we have seen that those severe patients who has recovered from COVID-19, there we find these neutralizing antibodies. So according to the team, including researchers from Tel Aviv University in Israel, some of the recovered patients tend to have robust and long-lasting immunity. Not all, some of the people. Why this disparity is there? The disparity is there because every human being has its own immune system, whether it is strong or weak, we don't know. So it all depends on the individual. So there are individual differences as well as the genetic factors also play a very important role in the immune system of every individual. So while others displaying a waning of their neutralizing antibodies, they said. Now, what are the types of vaccines against coronavirus developed till now? This is a very important thing which we should know. The first candidate in this competition is the live attenuated viruses. What are these? Attenuated means what? Attenuated means we are weakening that particular virus. So weakening a virus typically involves reducing its virulence. So reducing the virulence means we are decreasing its capacity to cause infection. So ability to replicate through genetic engineering, we will be doing this. The virus still infects the cell and cause mild symptoms. We are weakening the virus, but the virus has the capacity to cause mild infection. So for a live attenuated virus, an obvious safety concern that the virus might gain genetic changes. Now, see, the live attenuated virus we are using in a vaccine, but the concern is that that particular virus might gain genetic changes that enable it to revert back to more virulent state. So if this particular type of strategy is used, then it is also dangerous. So another worry is that a mistake during manufacturing could produce a defective vaccine and cause a disease outbreak. So these are the things which are to be taken into consideration before making any vaccine. So see what challenge the manufacturers are facing or the researchers are facing. So this same thing happened during the polio vaccine. So live attenuated vaccine based on SARS-CoV-2 are still undergoing preclinical trials or testing developed by startup. Now this has been tried by Codagenics and the Serum Institute of India. So these are the two companies who are working on this live attenuated viruses. So if these vaccines, live attenuated vaccines, if they are administered to the person who has impaired immune system, example, they have leukemia, or if they have HIV, or taking immunosuppressing medications like liver transplant, heart transplant, or kidney transplant, administration of a live attenuated vaccine might result in an uncontrolled replication of the virus. And this will be deleterious for that particular human being. So this particular concern has to be addressed before making a vaccine. Now, inactivated or dead vaccine. We have seen how the live vaccines are challenges. Then we have the inactivated vaccines. Inactivated means dead. So the scientists don't consider virus to be live. Now what they do? The virus will be the one you want to create a vaccine against, such as we want to create a vaccine against SARS-2, while usually killed with chemicals. Now what will we do? We will kill the virus with the chemical. Two Chinese firms, they are doing this. What they did? They have developed the vaccines that are being tested for safety and effectiveness in large-scale phase 3 trials. We have that. And they are CoronaVac, previously called PicoVac from Sinovac Laboratory, and New Crown COVID-19 from Sinopharm. These are the two vaccines which are using this particular technology of inactivated virus. So both drugs contain inactivated virus, didn't cause serious adverse side effects, and prompted the immune system to produce the antibodies against SARS-CoV. So Sinopharm's experimental vaccine has reportedly been administered to hundreds or thousands of people in China. And both the drugs are now being trialed in countries across Asia, South America, and Middle East. So inactivated vaccines usually require multiple doses. So this is the again a challenge because it requires multiple doses. Some inactivated vaccine may also require periodic supplemental doses to increase or boost the protection against disease. That means we have to administer it again and again. So this is the challenge. So these types of vaccine can be safely given to a person with an impaired immune system. This is a very good thing in this in case of this inactivated vaccine, because if the person is having a compromised immune system, he will not get the vaccine. But this will not happen if we have these types of vaccines. So this is a very good thing as far as the immune compromised patients like HIV or the patients like leukemia or the persons who are undergoing any immunosuppressive therapy. Then we have the recombinant viruses. Now this technology is very much used nowadays. Recombinant virus is a vector that combines the target antigen from one virus. Now here we have two viruses in combination. One virus with the backbone from another virus, unrelated virus. One virus against whom we are interested in making the vaccine, and the other virus which is unrelated. So for coronavirus, the most common strategy is to put coronavirus spike protein, the spike protein of coronavirus, into an adenovirus backbone. These adenovirus, we are very well known with this adenoviruses because they cause the common cold and many other symptoms in human body. So the recombinant virus are a double-edged sword. They are very good for us. Why? They behave like live attenuated virus. So a recombinant vaccine comes into with the potential benefit of provoking a robust response from the immune system, but also potential cause from causing an artificial infection that might lead to severe symptoms. Now, see, we can get the robust immune system with vaccination of recombinant virus, but the challenge is if artificial infection is created, then again we have to deal with that. So the recombinant vaccine might provoke an adequate immune response in people who have previously been exposed to adenoviruses. So if we are exposed to adenoviruses, then it will provoke an adequate immune response that infect humans. So the recombinant vaccines which are produced during this coronavirus are the CanSino Biologics in China and Sputnik 7 from Russia. So these are in the phase 3 of clinical trials and licensed for use in the military. So these vaccines, they have passed their third trial and therefore they are now in the market. So to maximize the chances of provoking the immune system, some vaccines are built upon viruses from other species. See, the same virus species, if we use, then we face challenges. So what the scientists are now doing, they are trying the other species. So human will have no pre-existing immunity because that, humans have not encountered that particular virus. So they will not have immunity against that particular thing. So in this category, the most high profile candidate is AZD122, better known as CHDOX1 and COV19, simply the Oxford vaccine, because it was designed by scientists at Oxford University, which will be manufactured by AstraZeneca. So this is based on chimpanzee adenovirus, not a human virus. It is based on the chimpanzee adenovirus. And it is seen to be 70% effective at preventing the COVID-19. So this is a very good candidate for coronavirus, what we can say the treatment or for vaccination. Some recombinant viruses can replicate in cell, others cannot be known as being replication competent or replication incompetent. So the vaccine candidate, one vaccine candidate that contains a replicating virus developed by pharmaceutical giant Merck is based on the this particular replication competent cells. And what they did is they infected the guinea pigs and they developed the vaccine. Then comes the protein vaccines. Now, these protein vaccines, actually, I'll give this. Okay, protein vaccines can consist of full length of spike protein. The name itself suggests. The name is protein vaccine. We know that virus is infecting us with the help of the protein, spike protein. So what the strategy is? The strategy is to take the spike protein as the key part, the tip of the spike that binds the ACE2 receptor on the cell surface, and ACE2 locks, and that a coronavirus picks it in order to break into the cell. So the ACE2 will bind to the spike protein, and in this way, the mode of action of this particular vaccine will take place. So manufacturing vaccine containing the protein alone has a practical advantage. How? Researchers don't have to deal with the live coronavirus. So there is no threat of the coronavirus, which should be grown inside cells within a biosafety level lab. Vaccine against only part of protein, a subunit, will be more vulnerable to being rendered useless if random mutation alter the protein. A very important, what you can say, thing which happens with the protein is that protein undergoes mutation. So the random mutation may occur in the protein, and that is the what you can hurdle in the manufacture of protein vaccine. So this random mutation protein, known as the antigenic drift, full-length proteins are harder to manufacture. So this full-length protein cannot be manufactured. The immune system can recognize either as an antigen. So this particular thing, if we manufacture, then it will be identified by the body as the antigen. So one candidate vaccine based on protein subunit is NVX-CoV2373 from Novavax, where the spike subunits are arranged as rosette structure. It's similar to the vaccine that's already been licensed for use. Flu block is already there in the market. So it is the Novavax is also the same type of protein vaccine. Then the vaccine development. So we come to the vaccine development. So development of a vaccine against COVID has taken place in unparalleled state. Usually such a process takes years. Now, because we are encountering the pandemic, we are in a haste to make the vaccine against the coronavirus. But the scope of pandemic triggered round the clock by thousands of researchers working over 100 different forms of vaccine all over the world. 100 different forms of vaccine are being tried. Effectiveness and safety were key concerns. So everybody, as a layman, we are also concerned that the effectiveness of that particular vaccine should be there, as well as the other things are to be taken into consideration. Pfizer vaccine approved in USA for emergency use has been found to be 95% efficient, and after its second dose. So this particular has been tested. Pfizer has already been tested, and it is being given to people. Before any vaccination can be used widely, it must go through the development and testing to make sure that its effectiveness against the virus or the bacteria, and it doesn't cause other problems. We have to see that. There are certain protocols. So the stages of development are generally like this. First is the exploratory stage. Now in this stage, what we do is, this is the start of lab research to find something that can treat or prevent a disease. So it all lasts 2 to 4 years. So we need 2 to 4 years for this exploratory stage. Then comes the preclinical stage. This preclinical stage, it lasts for 1 to 2 years. So scientists use lab tests and testing in animals such as mice or monkeys to learn whether a vaccine might work. So if the tests are successful and the FDA signs off, it's ready for the clinical testing. So after the preclinical stage, it goes to the clinical development. There is a 3-phase process of testing in humans. So if it goes to the clinical development, then we have to go through 3 phases. Phase 1 is usually 1 to 2 years and involves fewer than 100 people. In phase 1, we have fewer than 100 people. Phase 2 takes at least 2 years and includes several hundred people. Phase 3 lasts for 3 to 4 years and involves thousands of people. Whereas clinical trial process may stretch for 15 years or more. So to develop a vaccine, we need 15 years. About a third of vaccine make it from phase 1 to final approval. So 15 years are required for making a single vaccine. So regulatory review and approval is needed after this phase. Scientists with FDA and CDC go over the data from the clinical trials and then they sign off. And the last stage is manufacturing. So the vaccine goes into production. FDA inspects the factory and approves the drug levels. And then quality control is there. Scientists and the government agencies keep tabs on the drug making process and on the people who get the vaccine. They want to make sure it keeps working safely. So in this way, the whole process of development takes place. Now, vaccine storage. This is a very important aspect as far as the vaccines are concerned. Now, many vaccines have highly specific storage requirement. Now, the storage, I'll not elaborate much upon it, but I would like to mention certain things like the vaccines, majority of the vaccines, they are stored at the temperature of 2 to 8 degrees in the refrigerator and with a preferred average of 5 degrees centigrade. Though some should remain frozen in the range between -15 to -50. So therefore, Moderna and Pfizer vaccines, they are good for nothing in Indian scenario because they need these temperatures -15 to -50, and this cold chain has to be maintained. If we cannot maintain the cold chain, then the potency will be lost. So this is the real challenge in the Indian scenario. Additionally, many should be protected from light and are packaged appropriately as UV light can damage them. Live virus can tolerate freezing temperature and rapidly deteriorate once defrosted. So while inactivated vaccine more commonly requires stable temperatures, damaged at temperature extremes. So we have to see the temperature ranges and we have to see the cold chain maintenance of that particular thing. And there are many vaccines which are to be defrosted before they are administered to anyone. So it takes about 30 minutes in advance to defrost, and then only it will be given to the person in question. Now, a very important thing, India and COVID vaccine. Where are we? What we are doing? India's drug regulator has given green light to Covishield, the local name for Oxford AstraZeneca vaccine developed in USA, and Covaxin, locally made by pharma company Bharat Biotech. Now we all know that India is the vaccine powerhouse. It makes 60% of the world's vaccine and is home to half a dozen major manufacturers. So this is the scenario. Now, what vaccines we have manufactured? What are the things? Covishield is based on viral vector platform. I have explained you the types of vaccines being developed. Now, what is this Covishield made up of? The Covishield is made up of the viral vector platform. Now, what they did? A chimpanzee adenovirus called CHDOX1 is the vector that has been modified to carry the coronavirus spike protein into human cells. So the adenovirus vector platform for vaccine against traction during the battle to stop Ebola. This particular adenovirus was used as a vector when the Ebola has started. But in this case, we have used the chimpanzee. Now, this is true for Covishield. Now we speak about the Covaxin. Covaxin is based on an inactivated whole SARS-CoV-2 virion, an old platform that is also used to make polio vaccine. This is the old strategy, conventional form. So a virus disease producing capacity is inactivated. Under this method, it is inactivated vaccine, which means that it is made up of killed coronavirus. So the coronavirus is killed by chemicals or heat treatment or whatever the strategy is, making it safe to be injected into the human body. So the Bharat Biotech used the sample of coronavirus isolated by India's National Institute of Virology. When administered, immune cells can still recognize the dead virus, prompting the immune system to make antibodies against the pandemic virus. So 2 doses are given 4 weeks apart of both the vaccines, the Covishield and the Covaxin. And these vaccines can be stored at 2 degrees to 8 degrees centigrade. Now the number of doses. How many doses will we need? We should know all these things. Both Covishield and Covaxin have to be administered in 2 doses. Bharat Biotech's vaccine has been given approval for restricted use in emergency situation in the public interest as an abundant precaution. So see, this is very important. Emergency, the restricted use in emergency. This particular thing is very important because certain things has not taken place with this particular vaccine. So in clinical trial mode, which means consent has to be taken from vaccine recipient. So for this particular Bharat Biotech vaccine, that is Covaxin, you have to take consent from the person who is receiving the vaccine. The recipient will be monitored after receiving Covaxin shots. Dose interval, what will be the dose interval of these vaccines? The Union Health Ministry has ordered the administration of second dose of the COVID-19 vaccination to those who have completed 28 days after receiving the first dose. After 28 days, the second dose will be given. Now the delivery route, both Covishield and Covaxin are intramuscular injections. The age groups of beneficiaries, Serum Institute vaccine is approved for people aged 18 years and above, while Bharat Biotech's vaccine has been approved for those 12 years and above. There is no clarity on whether children and pregnant women can be given the vaccine. These vaccines, we don't know the protocol for the pregnant women and the children. Efficacy. Serum Institute of India vaccines at 2 full doses has been shown to have 62% efficacy in phase 3 trial. 2 full doses will be administered in India. Storage, as I already told you, 2 to 8 degrees centigrade. Manufacturing, both Covishield and Covaxin are made in India vaccine. Serum Institute of India has given an exclusive license from AstraZeneca Oxford University to manufacture and distribute the vaccine in India and other emerging markets. Now, a very important thing everyone is discussing, the vaccine controversy. What media says, these are not my views or not the views of the researchers. What the media says while interviewing the people. In June 2020, when the corona outbreak was there, when the pandemic was there, India's drug regulatory permitted chairman and managing director, Dr. Krishna Ila, Bharat Biotech International Limited chairman, to develop a homegrown vaccine in record time. Since then, the company has been buffeted by controversies, ranging from unrealistic government schedules to sporadic reports of adverse reaction. Matters came to a head last month after the government approved its shot before the completion of trials. That fueled a revolt across the breadth of India, with many of the frontline of the pandemic, particularly healthcare workers. They were more concerned, refusing the company's injection. So 2 weeks in, India's national vaccine rollout and fallen flat, with little over half the targeted number of people coming forward for shot. This happened with Covaxin. Hesitation that's largely being blamed on the hasty approval of Bharat Biotech's shot, which is still deep in phase 3 trial. Covaxin, now good news. After the controversy, the Bharat Biotech tried to prove their point. So Covaxin, India's first indigenous novel coronavirus vaccine, has demonstrated nearly 81% efficacy in preventing COVID-19 after the second dose in those without prior infection, according to its interim phase 3 trials. Results made public on Wednesday. This Wednesday. Today is Friday. So this is the report on Wednesday. This interim efficacy trend puts Covaxin at par with other global frontrunner vaccines. The Desi Vaccines co-developer, the Indian Council for Medical Research, said in a statement, the results evaluated by an independent data safety and monitoring board show that vaccine is well tolerated and efficacious against COVID-2 across a wide range of age groups and variants, ICMR added. So Covaxin also demonstrates significant immunogenicity against the rapidly emerging variants. And according to the Hyderabad-based vaccine maker, the inclusion of IMDG adjuvant. So this adjuvant has to be included in the vaccine. In which vaccine? This Covaxin. In the vaccine enhancer, it is the vaccine enhancer, and it enhances the T-cell immune response to COVID-19, leading to long-term protection. So the 80.6% efficacy figure was arrived at after 43 cases of COVID-19 were reported among the 25,800 phase 3 trial participants. So these are the results of Covaxin. Other 4 COVID-19 vaccines that are in pipeline in India are Zydus Cadila, Russia's Sputnik, Hyderabad-based biological BE vaccine, and Pune-based Genoa Pharmaceutical. All these are in the phase 2, some are in phase 1, some are in phase 3 trials. And this particular Pune-based Genoa Pharmaceutical vaccine is likely to be there in the market in the month of March. That is now. Now, what is COVAX? Why we are doing what good things, or we are helping the neighbors? And what are the strategies for that? COVAX is nothing but it's a part of access to COVID-19 tools, accelerator, a framework for global collaboration that set up in response to a call from G20 nations. We are in G20 nations in March 2020 at the start of coronavirus pandemic, and was subsequently launched by World Health Organization, the European Commission, France, and the Bill and Melinda Gates Foundation in April 2020. So this is COVAX is a you can say a collaborative approach. What is it for? This accelerator is made up of 3 pillars. What they do? Diagnostics, therapeutics, and vaccines. These are the mandates of COVAX. So COVAX pillars is co-managed by 3 partner agencies. Gavi. What is Gavi? Gavi is the Global Alliance for Vaccine and Immunization. Is a global health partnership of public and private sector organization dedicated to immunization for all. So we are not thinking about India itself. Since we are in the G20 nations, we are thinking about immunization for all. That is global immunization. So the Vaccine Alliance Gavi, the Coalition of Epidemic Preparedness Innovations CEPI, and WHO, it is only global initiative that is working with governments and manufacturers to ensure COVID-19 vaccines are available worldwide to both higher income and lower income countries. COVAX acts as a platform that will support the research, development, and manufacturing of a wide range of COVID-19 vaccine candidates, and will negotiate the pricing. By joining COVAX, all participating countries and economies, regardless of their ability to pay, because there are poor countries in the global scenario, will have the access to the portfolio of COVID-19 vaccines once they are developed and proven to be both safe and effective. So it is the work of the COVAX. According to Gavi, the COVAX is working. The initial goal is to procure and fairly distribute 2 billion doses of COVID-19 vaccine across almost 200 countries and economies by the end of 2021 through a mechanism, the COVAX facility. This COVAX facility is going to help the nations all over the world in the global scenario. That should be enough to protect high risk and vulnerable people as well as frontline health workers, according to Gavi. Now, what the WHO guidelines are for fair and equitable allocation of limited supplies. What the WHO says for the global scenario. Now, according to WHO, in phase 1, the COVAX countries initially receive vaccination. COVAX countries are there, G20 countries. So in the phase 1, this COVAX country, what will they do? Vaccination for 20% of the population. Enough to immunize target groups. What are the target groups? Health care workers, older adults, those with serious health conditions. This is the phase 1. Only 20% of the population is getting immunized. Then, in the phase 2, the pace of sending doses is prioritized by country risk. According to country, COVAX is going to decide phase 2. What they are going to plan? Countries under the risk of high impact of COVID-19, they will get the vaccines first. Then countries with vulnerable healthcare system. And then thirdly, countries with vulnerable population. So the African countries are there, Malaysian countries are there. There are many countries. Those are poor countries and they cannot afford to have the vaccines. So there, this COVAX is going to help. Then the humanitarian buffer. This is the last thing. The humanitarian buffer. In addition to phase 1 and phase 2, some doses of vaccine will be reserved for other vulnerable population What is this vulnerable population? It includes refugees, asylum seekers, and workers in these settings. So those workers who are working for the refugees and asylum seekers, for them, this humanitarian buffer will be reserved. So India is the world's largest vaccine manufacturer, ahead of even China. The Pune-based Serum Institute currently manufactures 5,000 doses of Covishield per minute, which adds up to 50 million doses per month. By the end of March, this figure will go up to 30%, which translates to 65 million doses. So India export scenario. So if time permits, I'll explain. Otherwise, I'll skip this because everyone knows what is the export scenario. Ma'am, should I go or should I stop?
Go on, but see the time limit, 5, 6.
Yeah. Exports, because we are proud of this export scenario because we are the exporters at large. Now, a significant part of coronavirus vaccines exported by India have been given as gift to the friendly countries. So, to whom we have given. So, between mid-January and second week of February, India exported more than 1.6 crore doses to around 20 countries. Around 62.7 lakh doses or 37% of the export to friendly countries were gifted. So here, the countries whom we have had the shipments are like UAE, South Africa, Brazil, Egypt, Morocco, Bangladesh, Algeria, and Kuwait. The Bangladesh purchased most of the vaccines, around 50 lakhs, followed by Brazil and Morocco with 20 lakhs. South Africa brought 10 lakh doses, while Kuwait and UAE brought 2 lakh each, followed by Egypt and Algeria with 50,000 each, as mentioned in the reports in Times of India. So this is the media report. And again, we are the suppliers to Nepal, Sri Lanka, Afghanistan, Bhutan, Maldives, Mauritius, Bahrain, Oman, Barbados with 1 lakh doses, Dominica with 75, and Seychelles with 50,000. So the health ministry said that vaccines are being exported after a thorough review of requirement in India. This has to be taken into consideration first. Before exporting to the neighboring countries, we have to see the requirement of our population. So it assured that India's needs will not be affected. So India has vaccinated 166,16048 so far, according to the latest data shared by Health Ministry on yesterday. This is the data of yesterday. And as many as 8516771 beneficiaries were vaccinated through 183,664 sessions. Through around 98,118 health workers have been taken their second dose too. India aims to vaccinate 3 crores healthcare and frontline workers in the first phase. These are the references. Thank you very much.
Best. You explained it in the very best way.
Very good.
Thank you so much, madam, for your valuable guidance in a very effective manner. So lecture is very nice and we come to know about COVID vaccine and all about the vaccine development. Now moving forward for question answer session, I request all participants to ask their queries, if any, to our respected speaker. You can put your questions in the chat box also. So participants are requested to ask their queries, if any.
Madam, I would like to congratulate you for this exhaustive account of the COVID vaccine, right from its biochemistry to its manufacturing to everything else. It was a wonderful presentation and really excellent presentation.
Thank you
for that.
Yeah. Thank you, ma'am.
And ma'am, especially the recent data which you have explained till now.
Yes.
It was best.
So I take
Thank you, ma'am. Thank you
I
once again.
really appreciate the information, and that very recent information you gave though. So it's really commendable. You did a good job.
Thank you, ma'am.
Yes,
Nice
ma'am.
lecture, Shalini. Yes,
Thank you
very
very
nice.
Yes.
I'm
Now
coming
I
to your
request.
Shalini
Right,
ma'am, there is a question we found in a chat box. If you
Yes, yes,
permit, can I read it for you?
Yes, sir. Yes.
The question is, can you suggest whether diabetic patients should be get vaccinated or not?
See, diabetic patient, actually, I'm not a physician to suggest whether you should take the vaccine or not. But as far as biochemical aspects are concerned, so being a biochemist, I can answer in that particular manner because only that will help. The thing is, diabetes doesn't mean that you cannot take the vaccine. When a person is suffering from the diabetes, we should first of all know what you say, the type of symptoms or the seriousness of that particular disease in that particular person. So if the diabetes is under control, then that person is as good as normal.
Yeah.
So
If there are
I
any
think
other complications like diabetic, if there are no diabetic complications, then the
then,
person
yeah,
is safe
there
to
is no
take vaccine. That is, I understand. That's what. Yes.
So,
Thank you, ma'am. And another question is still there.
yeah.
COVID vaccine is for lifetime or it is for specific time? That means the
No,
efficacy
COVID,
of
no, COVID vaccine is not for a lifetime. COVID vaccine is a very short, it has got a very short-term memory. And therefore, like all other vaccines, like for hepatitis and many other diseases, we take the vaccination again and again. Every year we have to take the vaccination. The same protocol may be followed in case of coronavirus vaccine.
Right,
Because it has got a short memory.
because that
You
is the
mean it
biggest
would be-
myth people are spreading nowadays.
No. You cannot have a lifelong memory for that. This is for sure, I can say.
One more question I found over here.
Yeah.
Taking vaccine increases your body temperature. Is it
Yeah.
good or bad?
It is good. Whenever any foreign substance enters your body, it is acting as an antigen. Whenever an antigen enters into your body, the body should react. And if your body is not reacting, it means that that particular antigen is not doing anything. It should not happen. So the body temperature should raise.
Right. Being a primary defense mechanism, body temperature must be increased.
Yeah.
One more question.
Yes.
The clinical trials of these vaccines taken within a year.
Yeah.
But if the side effects of these vaccines occurs after some year later, then what to do?
See, the Bharat Biotech vaccine, it has entered the third phase of clinical trials. Now we have to rely on them because they are saying that it is efficacious and it is 80% what you can say efficient to protect us from that the coronavirus disease. So considering all those aspects, we have to just believe them and we have to take the vaccine. And one more thing is that it is short-lived. We know that the effect of the vaccine is short-lived. So there is no question of harming the immune system in the long term. This is also one of the things.
Yeah, that's a good point. Yeah.
Yes. Now, I request Dr. Anuradha Gadkari Madam to sum up the session and share her valuable remark on today's program. Over to you, Anuradha Madam.
Thank you. First of
Gadkari
all,
Madam.
thanks.
Gadkari Madam.
Thank you. Yeah, thank you. Am I not audible?
I have,
Yes,
You
ma'am,
are.
you're audible.
yeah, you are audible. I need your permission to leave, madam. I have work. Can I please?
Thank you so much, Seema, for
Gadkari
everything
Madam.
you do for IFSA. Everything
Yeah,
you
yeah.
do for
So
IFSA,
please,
and you can. Yeah.
ma'am, please carry on, carry on. Thank you, ma'am.
Yeah.
Thank you.
Okay, okay. First of all, heartiest congratulations to Dr. Shalini for this well-studied, well-delivered, well-compounded, including every aspect in such a short span, in such a short. We are sorry we couldn't give you more time because it's difficult. And one thing, You kept everybody spellbound. We had no noise from the students from anywhere. And this performance of yours, hello?
Yes, you are audible, madam. Yes, please go on.
And this performance of yours, I can relate very nicely because you are my daughter's immediate junior, and that makes me very happy. And I can get from where you get all this from your department. Thank you so much. Another thing.
त्यांचं असं एक दोन मी मुलाखत काल संध्याकाळी
Please unmute the one who is speaking. Please, please
तर
unmute.
त्यांनी असं म्हटलं की आदिवासी समाजाचे पारंपरिक साधन
Mute your mic.
Seema ma'am and
आणि
somebody
हे
was
बांबूचे
speaking.
विविध
Please
स्वरूपातील
unmute.
वापरांमुळे
Please
आदिवासींचे
unmute.
जीवन सुकर होईल ही संकल्पना आपल्याला कशी काय
From
Dr.
where
Seema,
is this?
mute yourself.
त्यांनी त्या बांबूपासून वस्तू तयार केल्या आणि त्याचं
Please mute yourself.
तर ही कल्पना आपल्याला कशी आली की बांबू हे उपयोगाचं
Hey, from where is this coming? From where is this coming?
Seema Sumalar ma'am is speaking.
Dr. Seema, please mute yourself.
हो आणि आपल्या कार्याचे मूळ केंद्र
Okay,
मेळघाट
I'll
लवारा
message.
अतिशय
Ma'am, please
दुर्गम
make
स्थान
a
हे
call.
ठिकाण निवडण्यात
Give her a WhatsApp message. Give her a WhatsApp message.
Yeah, ma'am, it's done. Now it's done, ma'am.
Okay.
Yeah.
Okay.
Yeah, you can continue.
Okay. And another thing I would like to add is, if IFSA is completing 40 years and you joined IFSA nearly after 38 years, so you are, for IFSA, the youngest member, and you did so well. I clap for you for that. You are a perfect IFSite. Thank you so much. Now, I would say, I'm supposed to summarize, so I would say that Shalini did full justice to the subject. I mean to say that she's made it very interesting by telling the stories, telling different stories of Jenner and smallpox and all that, those interesting stories. She gave you very simple and basic answers to the basic questions, like explaining what is immune system, what is the vaccine, what is the history of vaccine, then what is antibodies. You know, these are simple questions which come to the mind of those who have not studied biochemistry. And you really explained it very well. What is the importance of vaccination? How does genetic factor come in the vaccination and in the immunity system? Then what are the challenges for the researchers? What are the different types of vaccines like attenuators, inactivators, recombinant protein vaccines? It was really very interesting. And you really gave the correct, you really touched the correct issue when you said that it takes 15 years to develop a vaccine. And here we are after 2 years. So this question is in fact is in everybody's mind. But that also you gave quite answers for that also. And that was very good. And finally, with dealing with what is co-vaccine was really good. Because many people have many questions, why the vaccines are going to the different countries when we do not have vaccines and all that. And what is our exportation and how, of course, this with the export, we are getting the goodwill of so many countries. That is also very important. You talked about herd immunity. That was good. You talked about spike immunity, herd immunity, and you also touched a very important point that the vaccine to be given, which vaccine to be given for the population above 18 and which can be given to the population above 12. So you touched all the important points and solved so many queries in a layman's mind about vaccine. And one thing, now we all of us are in dilemma whether to take the vaccine or not to take the vaccine. And I think this lecture of yours has to some extent or to a great extent has solved that issue. We have understood what is the vaccine, why is it necessary. And thank you very much. Thanks to you. And thanks to Kamala Nehru College, Principal Badwai, and all the students who joined you, listened to you without any noise, without any disturbance. And it was all went off so smoothly and so nicely. Thank you. And one request to Kamala Nehru College. be attached to IFSA. We come out with so many different activities for the college students, for the lecturers, and we would also request you to the teachers, more to the science faculty, if you can get associated with our activities. Recently, I had one Dr. Ketki from your college. We welcome her. Is she here today, Dr. Nisar?
Yes, yes, she is there, madam.
Welcome, madam. Welcome.
Now,
Yes,
Dr. Thakur can become a member.
Welcome.
Yes.
We are happy that you are going to be with us. We are happy that you are going to be with us with all this infrastructure in your college, with your principal support and all that. We look forward
Yes,
to
madam.
Shalini's College, Porwal College, and to Ketki's College, Kamla Nehru College, for all the cooperation in the future for IFSA activities. And your colleges are very important us because you are taking us to the rural area. And our target includes rural area also. For effective participation, it is necessary to have effective infrastructure in the rural area. And we look forward to your help. IFSA looks forward to your help and your effective participation. Thank you, sir. Thank you, principal, sir, and all the staff of Kamla Nehru College. Thanks to all the teachers. Thanks to all the students. Be with us always when we invite you. Be with us always when we invite you. That is my last words. Thank you very much.
Thank you, ma'am. Thank you, ma'am.
Yes.
Thanks. You have conducted the event very effectively and very nicely. So congratulations to all of you. Yes.
And claps for all those who conducted, who participated.
Yes, they conducted,
yeah. Clap, please clap. I'm not hearing any claps.
Yeah.
Thank you, madam.
Very nice.
So thank you, madam. Thank you very much. We definitely always will being associated with IFSA from day now. At the end, I request Dr. S.S. Wak sir, Head Department of Biochemistry, to propose formal vote of thanks. Over to you, Wak sir.
Thank you, ma'am. Very good afternoon to all of you.
Hello.
I'm Dr. Shardul Wag, and I'm here feeling very privileged to propose a formal vote of thanks on this today's wonderful lecture. At the onset, I pay my homage to great visionary leader, Honorable Late Shri Govindaraoji Vanjari sir, founder president of Amar Seva Mandal. At first, I'm thankful to Honorable Dr. Suhasini Vanjari madam, president of Amar Seva Mandal, and Honorable MLC Advocate Abhijit Ji Vanjari sir, secretary Amar Seva Mandal. So they are not to be here due to their busy schedule, but they are always with us as our constant source of inspiration. I sincerely thank to Honorable Dr. Smita Vanjari madam, treasurer Amar Seva Mandal. Without her guidance and support, it would have been impossible to arrange this event with such a short period. So thank you so much, ma'am. On behalf of entire institution, I sincerely thanks to convener of this popular lecture series, Dr. Pradnya Bhalerao madam. Thank you so much, madam, for letting us know more about IFSA and its contribution in various sectors. And definitely, as Bhalerao madam says, that Kamla Nehru College is definitely going to be one of the participants and associates for the IFSA and Nagpur chapter. I'm pleased to thank Dr. Shalini Chahande madam, guest speaker of today's wonderful lecture. Shalini ma'am, thank you so much for explaining such an important issue in a very easy way. I extend my thanks to respected Dr. Dilip Badwaik sir, Principal Kamla Nehru Mahavidyalaya, for his introductory remarks and ensuring the flow of all resources required to arrange this event. I'm also thankful to respected Dr. P.V. Dahikar sir, Vice Principal Kamla Nehru Mahavidyalaya, for his cooperation and coordination in arranging this event. I extend my thanks to Dr. Anuradha Gadkari madam, who had summarized this event so pleasantly. I'm very much delighted to extend my thanks to Dr. Sandhya Moge madam, Head Department of Biotechnology, Kamla Nehru Mahavidyalaya, for taking efforts to materialize this event successfully. My words of appreciation to Professor Neha Thakur madam, Head Environmental Science Department, for nicely comparing the program. Last but not the least, I would especially like to thank all the participants and students who have attended this national level seminar across the country. Because of your active participation, this program has been a great success. Thank you all. Now I request all the participants to switch on their camera for a group photograph. As we all know, pandemic guidelines are still there. So for a photograph, for the sake of photograph, please remove your mask so we can have a group photo. Can you please switch on your camera, all the participants? Nishant sir, if you're there.
Yes, sir, I am taking photo.
Yes, yes.
Just I am waiting for others.
Yes, yes, yes. And please let us know once you finish the photograph.
Yes, sir, I will inform to all of you.
Thank you, Nishant sir. Today's session is over now. Thank you very much for your participation. And it's a great pleasure, ma'am, to be associated with USA Nagpur chapter now.
You, sir.
We'll definitely take up
Thank you.
with a lot of activities. Thank you so much.
Thank you.
Sir,
Thank you.
please accept our congratulations as the grades you have received, your college has received. So I want to congratulate on behalf of USA. USA congratulates you, sir, for all your grades you received from NAC.